Okay, thank you, Alla. So as Alla said, my name is Jenna. I'm a fourth-year PhD candidate in Ben Wallazin's lab, and I have the pleasure today of working with Sara Rouhanifard today for our talk. Sara started her science journey at UMass Amherst with a BS in biochemistry and molecular biology. She then went to pursue her master's and her PhD in the lab of Peng Wu at Albert Einstein College, where she was working on developing magnetic glycomanoparticles and using histological methods to visualize complex polysaccharides in diseased tissue. She then went on for her postdoctoral work at UPenn with her mentor Arjun Raj, where she utilized high-power fluorescence microscopy to visualize RNA editing on mammalian cells using single-molecule RNA fish to study subcellular localization and single-cell heterogeneity. And now she's currently an assistant professor of bioengineering at Northeastern in our backwoods. And Sara has participated in many conferences and talks. She publishes in a lot of high-profile publications, and she has several patents right now on nucleic acid probes and devices related to nanopore. So please join me in welcoming Sara to the stage.
Thank you all for the invitation and for the kind introduction. Oh, sorry. Yes.
Can you hear? How's that?
That's good. Okay.
Very good.
Sorry about that. I'm an assistant professor in the bioengineering department at Northeastern. I started my lab in January 2019. And what the lab is really interested in doing is understanding how changes at the RNA level are going to regulate gene expression. So this is just kind of a lab overview. I won't talk to you about all of these topics today, but just generally what we're interested in. One half of the lab is understanding how mRNA modifications, specifically pseudouridine at the moment, are regulating gene expression. And we do three main areas for this half of the lab. One of them is using direct RNA nanopore sequencing to map where they are in the transcriptome. We also want to quantify them because, as I'll talk about, as the RNA is passing through the pore, it's producing current that we have to interpret. So the signal processing is very important. So one thing that we're doing is trying to interpret that signal so we can quantify occupancy of mRNA modifications. And then ultimately, this is all in service of figuring out the biological function of RNA modifications, specifically on mRNAs. So the other half of the lab is really more of a chemistry lab. I'm an organic chemist by training. So we're building tools for RNA detection. So as Jenna mentioned, I came from a single molecule RNA fish lab. So my dream is to be able to develop tools that we can site-specifically label mRNA modifications in situ. So one of the things that we're developing, and we recently posted on bioRxiv, is a copper catalyzed click chemistry method that works in live cells, intracellular labeling. And we're developing that so we can get mRNA specific labeling inside of live cells. And then we also have an area of the lab that's working on virus quantification using fish. But like I said, I won't talk about all of these things today. What I'll talk about today is RNA modification, specifically pseudouridine. So we have over 150 different types of RNA modifications that have been found through all classes of RNA, dominantly found in tRNAs and ribosomal RNAs. And the reason that a lot of these are found there is because these RNAs are highly modified, for one, and these RNAs are the dominant form of RNA that we find inside of our cells. By mass, that's the most RNA that you have inside of your cells. The mRNA is going to be less than 5% of the total RNA inside of your cells. But many of these are occurring on the mRNAs as well. So individual mRNA modifications, we know from a few well-studied examples that they can have extreme physiological consequences. One example that I like to use is A to I RNA editing. There's a single site in the transcriptome of healthy neurons that must be modified close to 100% of the time. So the A needs to be converted to an I in order for you to have a healthy neuron. So this modification being present prevents calcium influx into the cell. If that modification is not present, calcium enters the cell and it could lead to neuronal impairment. Now, we have other RNA modifications that we know of that occur on mRNAs, and this is not a complete list. This is just a handful of the ones that we're aware of, but every few months we'll find out about more and more. Very recently, at the beginning of this year, we added dihydrouridine to the list. So this is very much a dynamic field that as the tools are developing, we're learning more and more about the modification status of mRNAs in human cells. So we have all these modifications that are on mRNAs. We have a problem that we have very few mRNAs inside of a cell, and we don't really know where to look. Now, there are a few types of RNA modification that we've made more progress on. One of them that I mentioned previously is A to I editing. So the inosine modification, the reason that we know the most about this one is that when the A is converted to I by an ADAR enzyme, this leads to a change in the Watson-Crick base pairing. So once you change the Watson-Crick, it enables you to detect the modification by comparing your genomically encoded sequence to your cDNA sequence, and then you can find it. So it gives you something to look for. We also have the M6A modification that there was an enabling tool where there was an M6A specific antibody that was discovered. Now there are a variety of other tools as well, but when you have something that can specifically bind to the site, then you can do pull downs, or you can do an RT block experiment to figure out where the site is. This isn't the case for all of the other modifications. The other modifications, many of them are mass silent, so you can't detect them by mass spec. They don't do anything to the Watson-Crick base pairing, so it's really difficult to detect them. So the one that my lab is very interested in is pseudouridine, which is shown in purple right here. So pseudouridine, many of you have heard about it because it's a critical component of the SARS-CoV-2 mRNA vaccine. Now this is a 1-methyl form of the pseudouridine modification. And this 1-methylated form of the pseudouridine, it was discovered in vitro that when you add this modification, it helps the synthetic RNA evade an immune response once it's delivered to cells, which is great for a vaccine scenario because you want it to survive long enough in the cell that it can translate into a protein, which is ultimately what you want from your vaccine. Now, I like to show this as an example just because usually nobody's heard of pseudouridine, but now we have. So we know from in vitro models that this can have a really important physiological function. This is one of the most highly expressed mRNA modifications that occurs in human cells, and we really have no idea what it's doing inside of there, particularly on the mRNA sequences. So the little that we do know about it, we have some idea of what it could be doing, and a lot of these studies are correlative, not causative. So we know from the SARS-CoV-2 vaccine that it can suppress activation of the innate immune system. We also know that it's been involved in some RNA splicing, so you'll find it in your splice junctions. It has a dynamic response to heat shock, so it can be up or down regulated in response to cellular stress. It's been linked to some diseases as well, but we don't know exactly where they are. We don't know how many are there, and we don't know exactly what the contribution of them is to the biology. So these are sort of the 3 themes that I'll talk about during my presentation today. 3 major challenges in the field, specifically for pseudouridine, but this is also general for all RNA modifications. We need reliable maps. So when you're looking at the entire transcriptome, which specific positions do we expect to be modified? And can we say it with certainty? And this is important because the field is sort of littered with sites that we're not entirely sure of. So can we pick certain sites that everyone is in agreement for, that these sites are absolutely modified? The second theme is quantification. Once we find the sites, if a site is modified only 10% of the time, like if I have 100 transcripts but only 10 of them have a modification, is that as physiologically interesting as if I had something that had 100% occupancy? So if that site is always modified. We don't know the answer to that. We also don't know what the positional occupancy is most of the time. So it's critical that we figure out how to quantify these sites as well once we map them. And then finally, the most important question I would say is, what's the functional impact of an individual modification? Is it just happening stochastically because these enzymes are floating around and we have a bag of RNA that can just randomly hit at certain sites, or is it doing something that's there? I would say it's probably doing something because of the examples that we have of physiological impact, but this is the type of thing that we're trying to figure out. Now, the first point that I made is that the mapping is very important. Now, so far, a lot of the studies have been using Illumina sequencing, where you'll either use an antibody or some kind of chemical-based tagging method where you'll allow the chemical or antibody to bind to your RNA modification, and then you'll do a reverse transcriptase stop. So the reverse transcriptase will read, and then when it hits the chemical or it hits the antibody, it'll stop there. And at the site that it stops, you can assume that there's a modification there with appropriate controls. Now, using the Oxford Nanopore platform, instead of converting to cDNA, which is going to erase your modifications, you can use native RNA. So the native RNA is going to pass through the pore, and the pore is the blue part that you see there. And as it's passing through the pore, you're going to read electrical current as it's passing through. And this current is associated with a base caller that's going to extrapolate which specific base is passing through at a given time. Now, the signal processing here is the way of thinking about how to interpret the signal processing for RNA modifications. If a C is passing through the pore within a given sequence context, we would expect the current to look one way. Let's say that we expect the current to look like this red current that we're seeing here. And I know that it's messy, but just think about it as you're kind of seeing it go in a straight line here, more or less. But then if there's a modification going through, you're going to have a deviation in the signal. Oh, sorry. Thank you. So you're going to have a deviation in the signal current. Now, this is not straightforward to interpret, but you can see that there are differences there. And the base caller has been trained on canonical sequences. So the base caller can get screwed up when it encounters an RNA modification. And the reason for the base caller getting confused is because you have a different current that's going to occur for a different sequence context. So the negative controls when you're doing this type of experiment are really critical because you need some kind of baseline to connect your biological data to. So the two types of controls that I'll talk about today, one of them is called an unmodified transcriptome, where we create a matching transcriptome that we've erased all of the modifications. So then if you're looking at signal intensities, you can compare normal to, you can compare canonical nucleotides to something that has a modification on it and try to identify that way. Another way that you could use a negative control is using solid phase synthesis controls. So generating long RNA molecules with that recapitulate the sequence context of an mRNA, and you plant an RNA modification in the middle of them. Now, one of these can be extremely expensive, that one. This one is easier, but it has some caveats, and I'll try to highlight what both of them are. So we started by creating an in vitro transcribed RNA to provide the unmodified transcriptome. Now, the way that we do this is we start off, the experiment itself is going to be two libraries. So we'll have our native mRNA and then our transcribed mRNA control. The way that we generate this is we take our native RNA, we reverse transcribe it to cDNA, and then in vitro transcribe it into RNA. And when you do this, you're using canonical nucleotides in your IVT process, so you're effectively erasing all of the modifications that are on there. So you'll end up with a full transcriptome that matches your native mRNA, that all of the modifications have been erased. So then when you compare these two libraries, theoretically you can find where some of the modified sites are. So we're interested in pseudouridine specifically. And the good thing about studying pseudouridine is that it has a systematic base calling error that occurs at the site of the pseudouridine modification. So this is showing you two different genes that were validated in the literature for having a pseudouridine at these specific sites. So when we compare our IVT library that's on the bottom to two direct RNA libraries, you can see at the site of the pseudouridine modification, we have a U to C base calling error. So when it shows up as gray here, that means that it aligned to the reference sequence. When it shows up as a color, that means that it detected a mismatch. So we have this systematic base calling error that is different from standard nanopore error. This is sort of something that comes up a lot. People are very concerned about standard nanopore error being very high. Now, standard nanopore error is these other random colored blocks that you see here. It's different than the systematic error at the site of a modification. So we decided to take advantage of this U to C base calling error when we compare it to our IVT library. So we searched transcriptome-wide for sites that have significant differences between the U to C base calling error in the direct versus the IVT library. And we wanted to see, like, when we detect these sites, how do they benchmark against sites that have been picked up in the previous methods, Illumina-based methods, as having pseudouridine, so validated sites. So every dot on here was detected by nanopore sequencing using our algorithm, and then we compared it to the ones that are green or teal have been detected by nanopore plus one other method. The ones that are blue have been detected by two other methods, and three have been detected by three other methods. So I like to show this plot because in the RNA modification field, we don't have a ton of overlap between all of our methods, and that makes it really difficult as a biologist to know where to start. If your goal is to study pseudouridine biology and every paper is reporting a different site, at least we can look at this plot and say, okay, those pink ones are detected by four completely independent methods, four completely independent labs on the same cell line. So we can be pretty sure that those ones are modified. And you can choose. I mean, even the nanopore plus 2, these ones, and the nanopore plus 1, these methods are so different from each other that the probability of them picking up the same site is very low. So then we also applied it transcriptome-wide, and we found 1,691 targets that we identified de novo. And this is showing that the ones that have the highest error are part of the TROOP1 motif, which is a pseudouridine synthase that is the dominant one inside of a human cell. Now, a big question is, how quantitative is this method? If I show you something that here you have IDI1, which is our gene, and there's a specific position in IDI1 that has a base calling error that's close to 95%. Now, if I say something has 95% base calling error, does that mean that 95% of the transcripts have a pseudouridine on them? Probably not, but we wanted to prove it, so we generated these synthetic controls, and these are made by a combination of solid phase synthesis and in vitro transcribing RNA molecules. So if you remember the way that the pore looks, you have the pore and then your RNA is passing through it. So there's a certain region of the RNA that is sitting inside of the pore. So the region that's interacting with the pore is important for the way that the signal is going to get interpreted as the nucleotide is passing through the pore. So we weren't sure, and there hasn't really been a systematic study done to tell us how many nucleotides flanking the modification site are important for influencing the current signal. So we overshot the target. We generated 1,000 mer RNA sequences from sites that we picked up and that other labs have picked up to have a psi in them. We used solid phase synthesis to generate a 15 mer RNA that has a pseudouridine at that specific position that's found in the transcriptome. And then we ligated to 2 in vitro transcribed left arm and right arm. So this entire sequence is 1,000 mer, and we passed it through the nanopore. So we have an unmodified version and a modified version. So this is the modified version. If that base calling error were 100% quantitative, you would expect the stripe in the center to be completely blue, but it's not. It's partially red and partially blue. So that means the red is showing up as a U and the blue is showing up as a C. So that's one interesting thing, which we knew, but now we're proving it. The other interesting thing is this is 5 different sequences. So 5 different sites in the transcriptome that have their own synthetic control. The ratio of red to blue is different for every single one of them, which good to know, but bad for us when we want to generate synthetic controls because it costs a lot of money to generate these. And yeah, it's challenging. But now that we know, one thing that we could say is that when you use U to C base calling error, it's always going to undercall the psi. So it's always going to be something higher. So when I tell you that the U to C base calling error for IDI1 is 95%, it's something greater than or equal to 95%. So we can actually look at it. It's kind of semi-quantitative in the sense that I can say that one is very highly modified. It's almost 100%. I can't tell you what the actual number is, but I can say it's above 95%. So then this is the same plot I showed before. I just compressed it a little bit. So now when we're looking at this U to C mismatch error, and then this is looking at the log of the transcripts per million, we made an arbitrary cutoff at 40% U to C base calling error and said that everything above 40%, we can consider these hypermodified or very highly modified by pseudouridine RNAs. And the motifs that were showing up are TROOP1 motifs, which makes a lot of sense. This goes with what we found in the literature, that motif is a pseudouridine synthase motif. So I've just spent a lot of time telling you that the method is not quantitative, but we can extrapolate some biology from it because maybe we're interested in starting with the very highly modified sites. Those might be the most easy to go after first because maybe the phenotypes that we observe will be the most extreme for ones that have been converted almost 100%. But we also wanted to see, since we have these synthetically generated controls, we have the unmodified version and the modified version, can we use machine learning to train on those sequence-specific synthetic controls? So we did that for those 5 targets that we generated. And something interesting that we found, so previously I showed you that all of these ratios were different, and this is completely based on the base caller. We don't know exactly what parameters, what signal features the base caller is using because it's proprietary. But we used all of the signal features that we could think of, and we extracted 45 signal features, and then we did a comparison, and we trained a machine learning classifier on these sequence-specific synthetic controls. So what we learned, we rank ordered all of those features. This is 45 total features that we trained on. We rank ordered the ones that were most important for classifying the pseudouridine correctly. Now this is just for 2 of them, just as an example. So when we rank order them, this is telling you that the feature is the K-mer, so the 5-mer sequence where the center one is the modified nucleotide. In this case, the number 1 that had the most weight towards identifying the psi was the K-mer quartile 1 at the 0 position, so at the modification position. Now for MCM5, this is a different one. The most important feature was the K-mer at the +2 position. So the modification plus 2 nucleotides to the right in quartile 1. So that tells us that it's different features for every single modification site that are going to contribute to identifying the psi. Now, again, this is bad news for someone who wants to generate synthetic controls because it suggests that you have to generate a new synthetic control for every single sequence target that you're interested in going after. And this is the topic of a new grant that we're working on right now. How can we make that more practical? If this is the way that you can achieve absolute quantification, then how can we get around the problem of RNA synthesis not being so cheap, accessible, and long? So we took this machine learning classifier and then applied it to 7 different cell types, many of which we sequenced on our own. some of which we took from public databases. This is the great thing about nanopore sequencing data. All of the current signal is published. So you can extract it and then reanalyze it forever. So we took some of these and then we applied our machine learning classifier, and we were able to get absolute quantification only at these 5 sites that we had synthetic controls for. So this is something that we're really interested in doing moving forward. For the sites that we do have controls for, can we quantify them and try to extrapolate the biology from it? So in my next set of slides, I'm going to try to give you a flavor for how we're now shifting over to learning about the biology. Now that we have a method that we can map, we know how to quantify, it's expensive and we're working on it, but now we want to use that with what we have and conservatively try to learn some biology from it. So this is a different method, another single molecule method called single molecule RNA fish. So this image you're looking at here is a group of cultured mammalian cells. So each of these circles is one cell, and each of these dots that you're looking at here is a single RNA molecule. So the method is called single molecule RNA fish, so fluorescence in situ hybridization. The way that you achieve this single molecule resolution is you'll design DNA probes, and each of these is about 20 nucleotides long that have a fluorescent dye on them. And you'll tile 20 to 30 probes across your target mRNA sequence. And each of them has the same dye attached to them. Now, the question is always, why can't you just use one probe? Why do we use 20 to 30 probes? And the answer is a 20-mer DNA is going to bind non-specifically all over the place on the cell. And for fish, we rely on localized amplification of 20 that are flanking each other on the mRNA target to achieve the single molecule resolution. So then you take a stack of 30 slices through your cell, and then I can tell you this one has 234 spots in it, and that represents one mRNA. This one has 562. I'm making up numbers. So there's things we can say, and then this one is not transcribing, neither is this one. This one's very lowly transcribing. So we can get a lot of information about the RNA, specifically about localization, and then single cell expression when we use a technique like RNA fish. Now, when we're using 20 to 30 probes that are tiled across your mRNA sequence, that's not really useful when you think about it in the context of a single modification in the center of your mRNA sequence. We need other methods that can help you get single nucleotide specificity, which our lab is working on, but I'm not going to talk to you about that specifically today. Another thing you can do, and we talked about this on a previous slide, some of these targets have very high U to C error, like greater than 90%. But even, I would argue, the ones that are lower, the ones that are down here. So everything that we've defined as high occupancy, even though we don't know what the exact number is, one could say something like most of those transcripts are going to be modified. So what if we use single molecule RNA fish and only try to image the ones that have very high occupancy? And we make the assumption that every spot we look at on there is probably modified. Then we can start to look at single cell expression and start to look at localization and start to get some kind of information about what might be happening at the RNA level when there's a modification present. And this is something new in the lab. We've just started doing it recently. So this is very preliminary data from the lab, but it was kind of cool already. So these are 3 targets that I grabbed from the hyper-modified list that I showed you previously. And we've just done single molecule RNA fish on wild-type HeLa cells. And also we knock down the enzyme TRUB1 that's responsible for generating those modification sites in the cells. So we're just doing plain vanilla fish on these. So every dot represents every single one of those mRNAs inside of the cell. So for AK2, you can see that the localization looks pretty similar in the wild type and the TRUB1 knockdown. When we switch to PFKP, it kind of looks like these ones have these bright spots in the nucleus. Maybe these ones don't have it as much. And these bright spots are called transcription sites. So this is the site where your polymerase has plopped down on your DNA and it's chugging out a whole bunch of RNAs at once. So you end up getting a bright spot in the nucleus that represents a burst, a transcriptional burst. And then with GT3, you can see something similar as well. We have these very bright transcription sites, and then when we knock down the TRIB1 enzyme, the transcription site seems to disappear. Now, we don't know how to interpret that yet. It's just an observation that we made for now, but this is how we might use RNA fish to look at some kind of phenotype from an mRNA modification being present. So some of the things we're interested in doing is looking at translation rate, decay, localization, again, transcription activation. So presence of the transcription site in the presence of and absence of the modification. So going back to the slide that I showed you previously, we generate these unmodified transcriptomes using in vitro transcription. So just briefly to go over it again, we take our native mRNA library, we reverse transcribe it to make cDNA, and when you make cDNA, you effectively erased all of your modifications. Then you in vitro transcribe it to create a transcribed mRNA control. So all of the modifications have been erased because you're using canonical RNA nucleotides at this point. So then you have an in vitro transcribed library that's a baseline that you can compare everything to. So any deviation from that means that you have a non-canonical nucleotide. And for Psi, we're detecting it using the U to C base calling error. Now, this is not the only modification that this can be used for. This one just happens to have the strongest base calling error signature. The other ones like M6A and other methylated nucleotides, you can also use this for, but the base calling error is not as strong. That doesn't mean we can't do it, but base calling error today is the easiest way to interpret RNA modifications on nanopore. The signal level stuff is in development. I would say, I think most people would agree with me that the signal level analysis is not ready for prime time yet. The base calling error is what's most commonly used today and the most practical way to look at something transcriptome-wide. So the reason I brought this up again is because a lot of people ask the question, we did this first study using HeLa cells. A lot of people are interested in studying RNA modifications in human cells, but not necessarily HeLa. So I was getting a lot of questions like, can we use your published IVT for any human cell line? And there are some maybe obvious, maybe not so obvious reasons why you can and you can't. So the answer is yes and no. So can a single human IVT control from one cell line be used for all human cell lines? So we posted this very small study on bioRxiv recently trying to describe how one human cell line can be used, the upsides and downsides, and how expanding to multiple cell lines and pooling them can be beneficial. So what we did was we created IVT libraries for, there's only 5 here, but we actually did it for 6 human cell lines, representing a variety of different tissue types. So we have lung, HeLa, liver, testes, and neuroblastoma. So a wide variety of different types of cells. So the single nucleotide variants are going to be cell type specific. And if we don't have a correct annotation of those in your IVT control, then you can misinterpret a mismatch in your direct RNA sequencing as a modification. So if the HeLa cell doesn't have the SNP, and you're comparing it to an SHSY5Y that does, then you might misinterpret it as a modification when it's actually a variant that's occurring at the genomic level. So the other thing is the mRNA expression, and I know this sounds obvious, but the mRNA expression is going to be cell type specific. So from a single cell line, you might have insufficient coverage. So what we did in this study was we produced 6 different cell types, IVT libraries for all 6 as a resource. And these are kind of the most commonly used cell types for RNA modification research today. So the hope was that if somebody needs to access this, this removes a barrier to entry. You can just download the FAST5 files that we've provided. But also there's some power in combining all of those. So we made a kind of a pan-human IVT control that has much higher coverage than what we get from a single cell line. And it also, we created these .bed files that will outline where a putative SNP is at different thresholding. So you can choose what kind of threshold you want. So if the SNP is only present at 20% of your reads, you can choose to be extremely conservative and have that show up when you're looking at your reads on IGB. So it'll tell you, like, here, maybe don't look at that site because it's shown up as a SNP on one of the other cell lines. So we tried to create this as a resource because using Nanopore for RNA modification analysis, I think it's the future of how RNA modification analysis is going to be done, but there are some barriers to entry, I would say. So we're hoping that this sort of decreases some of those barriers to entry. So another thing that we did, so we generated those IVT controls for 6 different cell lines, and there are some really simple questions that nobody's really been able to ask before. So one of those questions, I know there's a lot of colors on here. This is very new data. I got it like at 11 p.m. last night. So this is, we did the sequencing and the Psi analysis for 6 human cell lines. We just wanted to ask the question, which cell line has the most modifications? Or which cell line do we think has most Psi modifications? And I still can't answer that question for you, but I can tell you some other things about it. It turns out that making a conservative call like that, we have to be very careful with how we filter the data. But here I'm showing you something really preliminary that we think we've been extremely conservative with. So here we see you have 6 human cell lines, and these are all of the Psi positions that met our criteria. And one of the criteria that, there were many criteria, I'll tell you one of the important ones is that the position that the Psi is in has coverage in all 6 cell lines. So it's not that the gene is not expressed, it's the gene has to be expressed, and it had to have at least 30 reads by direct RNA sequencing in order to make the list. So we wanted to be able to see if one cell line has the target sequence and not the modification, and then compare that to another one that has the sequence but has the modification. So the way that we sorted it so far, and we're still playing around with this, is up top here, these sites only showed up in 1 cell line and not in 5. The ones that say 2 showed up in 2 cell lines and not in 4. 3 showed up in half, and so on and so on. So we're still digging into this. I think the most interesting ones to focus in on are the extremes. So the 6 means that the site showed up in all 6 cell lines at variable levels. And we're doing an analysis of that, which I'll show you in a second. It's also interesting finding the ones that have just 1. Particularly to me, the ones that are shown in pink and orange, because pink and orange has the highest base calling error. And something that we've noticed as we're doing this comparative analysis, if it's high in 1 cell line, it tends to also be high in other cell lines, which you can kind of see here. So these ones are expressed in all 6 cell lines. And it's kind of interesting that you have this orange region. That means that these are almost 100% modified in all of the cell lines, except for the neuroblastoma cell line, which is also kind of high. That one's 60 to 69% UTC base calling error, which I've shown before is undercalling the sites. So we're still trying to interpret this, but we're moving in the direction of figuring out what these are doing there, which sites should we be putting all of our energy towards for analyzing. So this is starting to give us some hints. Maybe these sites are super interesting because they have to be 100% modified, and they're 100% modified in all of the cells. Other interesting observations that we made. This gene right here, DK1C, that one is kind of interesting because I haven't shown you knockdown data, but we've done it, and we know that this particular gene at this site is modified by TRUB1, which is another pseudouridine synthase. DK1C is also a pseudouridine synthase. So this is an example of one pseudouridine synthase modifying another one with pseudouridine, which is kind of interesting. I don't know what it means, but we're just starting to make these very interesting observations that we're starting to flesh out. So another area that we're working on, like I said, these are all kind of new. I'm just giving you a flavor for how we're trying to figure out what the biology is. So in order to figure out the biology, a lot of times you need to apply some kind of perturbation to the system and see how it changes. So what we did here was we took one cell line, SHSY5Y cells, and we did a retinoic acid differentiation. So this makes them neuron-like. It gives them these long processes coming out of them. It changes the gene expression profile. So it's changing the cell state. We're using it as a model of changing cell state. So there's a major morphological change that's occurring and also like a gene expression change that's occurring, a major one. The other thing that we did was we treated the SHSY5Y cells with lead. So we poisoned the cells. So two very different types of cell state change. One of them is sort of a more natural process, and the other one is like a probably more cellular stress for this one. So we wanted to see what are the differences in pseudouridine sites for these two different types of changes for the same cell type. So we compared our undifferentiated cells, the Psi sites that we picked up in the undifferentiated cells to the differentiation profile. This is a comparative analysis. So everything that you see in this region right here is not really changing much. But when you look at things that are in the white regions, this means that in response to differentiation, the Psi percentage went up. Down here, it means that the Psi percentage went down when we compared to undifferentiated. And then we also did a trend line analysis when we compared to two different motifs, the two most common for humans, PUS7 and TRUB1. And you can see that in response to differentiation, the PUS7 motif, or the sites that fall within a PUS7 motif, are decreasing. So then we did the same analysis for lead-treated cells. The trend lines were not as significant, but you can see there's different sites showing up. So then we wanted to compare the two different types of perturbation to each other. And this truly is something that is like very much a work in progress. We're even still figuring out the right way to plot it, but this is the one I like the best so far. So this one, we're comparing the difference between lead-treated and undifferentiated cells to the differentiated. So everything that you see in the center of this, that kind of means that for all three conditions, that means lead, untreated, and differentiated, the Psi modification is stable. It's the same for all of them. Now, for this line that's going through, everything that falls along that line, it means that the change is correlated, sort of. So it means that for this condition, if you're getting 10% less Psi in response to lead, you're also getting 10% less Psi in response to differentiation. And then we also have other interesting things showing up where you have all these outliers. But this really is something that we're playing around with now. It's starting to look interesting because you can see that there are certain modifications within human cells on mRNAs that are stable, which is interesting in and of itself. And there's other ones that are very highly dynamic. So we're trying to take sort of a systems level approach to try to figure out what the differences are between those two, and we're trying to flesh it out. So far, it's really just trying to interpret those plots, but I just wanted to give you a flavor of the types of questions that we're starting to ask. So then, just to close out, we had three different areas that we're interested in and the field of RNA modifications is interested in. So one of them is mapping. So our approach is to use direct RNA sequencing and compare to unmodified transcriptomes. And then quantification, really the big area for us is the development of synthetic controls and the machine learning algorithms to try to quantify specific sites. And then biology, this is the area that we're very much expanding on right now, but we're really at the beginning of it. So figuring out evolutionary conservation of the sites across species to try to figure out which ones are the most important, looking at variability across cell lines, and then dynamic response to a lot of different types of perturbation. And then I'll end there and see if anyone has any questions. And thank you guys for listening.
Moderate the questions.
I have two questions. One about pseudo-regulation. Did you try to localize organelle pseudo-regulation? Mitochondria, nuclei, cytoplasm, and what state cell growth? So why?
So we are not looking at mitochondria, although mitochondrial RNAs do show up in some of our hits. So you mean localization by fish?
No, just by
Fractionation.
yes, the second second question.
Yeah, so we haven't done fractionation. So we're looking at whole cells for all of our studies so far. And
Do you think it's possible?
to fractionate and do it? Yes, but you would need a lot of cells. Well, a lot. So the current chemistry for nanopore requires 500 nanograms of mRNA, and it's tricky to get enough when you're fractionating. You need to scale up quite a bit. It's not a big deal for something like HeLa cells, but for other types of cells, like smaller ones, like germ-cat cells, for example, we have to scale up quite a lot to get enough RNA to do these studies with.
And my second question about fish. What is the smallest RNA? Right, because you said that you used 20 probes to distinguish one RNA, but small RNAs are abundant. Can you prove it?
So
With confidence.
yes and no. So one could do a different type of fish where you make the probe longer, and that would have a higher probability of hitting your target sequence. You're a little more limited in the types of controls you could do, because imagine you have a long RNA that you can design fish probes for. Let's say I design 30 fish probes for it, and I'm getting spots. And you can make the assumption that those spots are what you want, or you could do this experiment called odds evens, where you split your probe set in half, and every other probe you give it a different color, and then you localize those. So you would be limited in your controls when you limit the sequence. The other thing is you don't always need 20 to 30 probes. There are probe sets that I've worked with that it's a full-length mRNA, so a normal size, like 1,000 nucleotides or whatever, 1,500, and a probe set of 6 was sufficient to give really bright spots. We don't know how many of those 20 to 30 actually bind. 20 to 30 is probably overshooting, but we don't know which fraction of those 20 to 30 actually bind. So I think you're asking about something that's under 100, right? I would say maybe. It's worth trying. Sometimes you just don't know, like something's hybridized better than others. If you want it bad enough, you can play with the salt conditions and the formamide conditions to force it. But,
A lot of the times in your quantification, you're looking at sort of at the gene level. And I'm curious how you account for differential pseudorelation of like some sort of transcript, right? Where one side's up and one side's down. You're getting
what do
mixed
you mean
modification.
We're getting less quantification?
Mixed modifications.
If there's another modification, not
so
just psi.
Right. Well, no. Even if psi is not necessarily happening a single time in a transcript, right? You're getting multiple pseudorelation events sometimes in a transcript. So how do you account for if one site goes up but another site on the same transcript is decreased or something?
Okay, that's a good question. The proximity of the modifications on mRNAs, they seem to be much further apart, like hundreds of nucleotides apart. We've seen very few that seem to be very close to each other. That happens a lot in ribosomal RNA and tRNA, that they're very close proximity. But when they're that far apart, we wouldn't expect them to affect the current of each other. Now, this is also operating in a black box that we don't really know what's on the sequence. So it's possible that there's some modification there that we don't know of. So at this moment, I would say you do the best you can for sites that look clean around it, and you do as many controls as you can. So if everything looks the way you expect, then those are the sites that we can interpret, but we can never rule out an unknown modification that's in close proximity. It would affect the signal, absolutely.
That was great. And then see, you started looking at trying to see which cell type has the most RNA modifications. What about tissues? Have you tried looking, comparing brain versus muscle versus lung? Could you get total RNA from tissues and run them through the nanopore? Is it too many cell types and that makes a bag that would complicate it?
No, it's a good question. It's something that I want to do, but I want to have a better handle of how a single cell type behaves before doing that, because everything that we develop, we have to do it from scratch. We have no toolbox. So I think for making some kind of biological inference, it's easier to go after a single cell type. Now, another thing that we're doing that I didn't mention because it's like fresh off the sequencer type of thing, but there's no primary human cell line that has been studied for RNA modifications using nanopore sequencing. So we did this extremely expensive experiment where we got primary human T cells, like purified CD4 T cells, and then we're doing the analysis on those. So we're working our way towards tissue level, and the first step is doing an analysis of primary versus immortalized, and then after studying the differences in that, then shifting over. But I don't know if, as a field, we have a good enough handle on how to analyze the nanopore data to switch into a more complex tissue yet. I think we're close, but I personally wouldn't do it yet.
I see. Okay.
Yeah.
Just to make things even more complicated, do you think it's possible that different cells within a population may differ in the modification of a given mRNA, depending spatially, or particularly in neurons in the brain, for instance?
Yeah, absolutely. So I also didn't talk about this, but like this is looking at just normal single molecule fish. Now, if we have 50% occupancy at a given site, one of the tools that we're developing is so that we could site-specifically label which one is modified and which one is not. And a big hypothesis in the field is that a modification occurs to drive localization. It's a hypothesis. Nobody's really proven it except for A to I editing. There's a localization hypothesis that has some basis in localization in neurons. But so it's definitely something that I think we will see variability. I don't know if it'll be localization or something else. It might be single cell variability, but yeah, I think the imaging tool is going to give us a lot more information about the biology, but such a tool doesn't exist.
I have a question about the fish assay where you looked in the TRUB1 mutant, and in some RNAs it was more accumulated in the nucleus. So I was wondering about sort of genetic compensation. I was wondering if you've done the nanopore sequencing in the TRUB1 mutant. So like some RNAs had an effect and others don't. And I was wondering, like, do you know how much of the pseudouridine changes when you knock down TRUB1?
So how much of the pseudouridine or how much of the enzyme?
No, so I'm wondering, does TRUB1 account for all the pseudouridine that you see, or are there other pseudouridine synthases that could compensate in the absence of that enzyme?
Yeah, it's only a fraction of them.
Okay.
And it's the fraction that is the highest percentage UTC error. So, and this is known in the field. So it doesn't account for all of them. Many of them are also +7. Those are the two most common in human cells, but there's 12 others that we know of.
Okay.
So yeah, everything that we do is just a fraction based on some control that we had at the time.
Is anyone looking picking up on what you were saying about localization, is anyone looking to select mRNAs? You know, knowing something about their functional localization. One that comes to mind because in my past would be actin, because Robson first identified the zip code directing them to a certain location in the cell. So of known mRNAs, is there something you can target, you can modify and demonstrate with fish that you lose that localization, or that situation is not available yet?
So the only example of looking at pseudouridine specifically, but other modifications using fish is kind of what I showed you today. Nobody's really done it. And I think the reason that we're doing it is because we're making a big assumption. You can agree or disagree with it, right? We're making the assumption that if we go after the highly modified ones, all of those are modified. So I guess your question is, can you go after one that you assume has a localization change and then work backwards to figure out if there's a mod there? We haven't done that analysis specifically, but we would have to get kind of lucky with that target because we picked up 1,600 sites in HeLa cells, which is not that many when you think about the size of the transcriptome. So I think we would have to get very lucky, and it would also have to be a high occupancy site. So not only would it have to be present on that transcript, but also like close to 100% to do it by this method. Now, if we had a way to figure out which one's modified, which one's not, then something like that I think would be feasible. But yeah, the method doesn't exist.
Are there any sites known where they can be the same position can be modified in different ways? Like, you get M6A. I know M6A goes to M6AM, but M6A or another modification of adenosine or you know what I mean? Or the pseudouridine, another modification of it.
It would be an amazing discovery, and it's possible. It sounds like something that probably will turn out to be true, but we have so few tools to look at even one that I wouldn't even know where to start to answer that question. But the cell does weird things. I feel like that sounds like the type of thing that it could do. And
for a lysine, you have that. Acetylation, ubiquitination, very different things.
Different things, but all of these processes sort of parallel each other in what they can do. So I wouldn't be surprised if such a thing was discovered. It's just that the field is so new in figuring out where everything is that I wouldn't know how to even start doing such a study.
But the inosine comes from adenosine, right?
Yeah.
So I could look for a cross-reference your inosine site to your M6A sites.
Potentially, yeah.
Okay, thank you all so much for your attendance.